welcome. And so Gabriela Merino has a PhD from the University of Córdoba in Argentina. She has had a postdoc position at the Zinc Institute in Santa Fe in my lab. And now she's recently entered CONICET as assistant researcher at the Bioinformatics and Bioengineering Institute of the University of Entre Rios, Argentina. Her research interests are machine learning, genomics, and transcriptomics data analysis. And she will talk today about deep learning models for predicting protein functions. Please, Gabi, go ahead. You can share your screen.
Okay, thank you, Georgina, for the presentation. Please let me know if you are looking at my screen.
Yes.
Yes? Okay, I will start the presentation now. So thank you for the introduction or for inviting me for presenting my work here. It is a pleasure for me. So the agenda of my presentation is here. So I will try to introduce myself in order to letting you know a bit about my previous work and not only my work, but also the collaborations that I have the opportunity to have with different women doing research on bioinformatics. I will then try to introduce you on the problem of automatic prediction of protein functions. Then I will try to present some of the state-of-the-art tools, and then move on our proposal of deep learning models integrating data from multiple sources for predicting protein functions. And then I will present you some of the ideas that we are planning to do in the future. So let's start about my introduction. As Georgina said, I am from Argentina. I am a bioengineering. Then I did a master on applied statistics and a PhD on engineering sciences. And now I am a postdoc with a transition to become an assistant researcher from CONICET. During my master, I had the opportunity to work with two great researchers from INTA CONICET, Norma Paniego and Carla Filippi. I have been working with them in the development of an R package for the imputation of missing genotypes in GBS data, mainly based on machine learning models, more specifically on random forest. And during my PhD, I have also collaborated with Andrea Lera from Instituto Leloir in the development of an R package for quality control of targeted sequencing experiments. This package is called TARSEQ-QC, and you can find it on Bioconductor. But the main aim of my thesis, my PhD thesis, was to develop bioinformatics tools directed to analyze and the study of alternative splicing. So in the context of my PhD thesis, I had the opportunity to work with one of the women leading the bioinformatics research on the world, I think, that is Ana Conesa. I had the opportunity to visit twice her lab in Valencia. And after the work that I started with Anna and my PhD supervisor, I developed another R package that is also available in Bioconductor that is aimed to analyze differential splicing using RNA-seq data. But then when I started my postdoc, I moved from the RNA-seq data analysis or NGS data analysis to the development of intelligent algorithms for inferring protein functions from data integration. As Georgina said, she's my supervisor, so I am working under her supervision, and Victor Casco from the Research and Development Institute on Bioengineering and Bioinformatics is my co-advisor. In the context of my postdoctoral project, I had the opportunity also to visit the EBI-MBL as a Cabana Secondi. I have been during 8 months under the supervision of Maria Martin, who is the leader of the Protein Function Development Group, which is part of the UniProt consortium. So about the problem that we are trying to figure out today, I will start a bit about talking about how are the protein functions represented. Elizabeth was talking a bit about Gene Ontology, and Gene Ontology is the most used way for representing protein functions. It is a framework of controlled vocabulary that is describing 3 different aspects of gene products function. Biological processes that are describing, as the name says, biological processes in which proteins are involved. Cellular components that are not aimed to, it is not aimed to describe protein function, but it is used for describing the locations of proteins or the locations in which proteins are carrying out their functions. And a molecular function that is aimed to describe the functionalities that proteins are carrying out as a complex. So each of those can be thought as an independent ontology or 3 independent sub-ontology of Gene Ontology, with the characteristic or the particularity that each of those ontology is implemented as a directed acyclic graph. So each node is representing a term. So you can appreciate here, for instance, the name of the term, that is this Go 0071704, and the description of the term that is telling us about what is this term representing. And in the graph, we have the more general terms in the top of the 3 or in the top of the graph, and the most specific terms in the deepest part. So each branch also in the graph is telling us about the relationship that we can find or we can identify among the different terms. In this case, the relations are all is a relation, so this node is a child of this node, but we can identify another kind of relationships, that those that were explained before by Elisa. So if we, for instance, look for a particular gene product in the Gene Ontology, we can obtain a table like this. So we have information about the protein or the gene product, such as the name or the gene symbol and the UniProt identifier. We can also obtain information, of course, about the annotation of the Go term that are assigned to this protein. Of course, we should obtain, or we should also know if these terms belong from protein, from biological process, cellular component, or molecular function ontology. We also can obtain information about the source of this annotation, and an important point is the information about the evidence that is supporting this annotation. That is a way for telling us about how reliable this annotation is, because the annotation can be obtained from an experiment, for instance, a biological experiment, or it can be inferred by automatic annotation tools. So why we need automatic annotation tools for predicting protein functions instead of only using data from experiments? Well, the thing is well illustrated, at least for me, in this plot that you can see here. This graph is from this paper, you can then explore it, but it is illustrating the number of sequences that were deposited in UniProt KB over the past decade. So as you can appreciate, there is a big gap between the number of sequences that are deposited. So they are sequences that we know the sequence of the protein, or we know the sequence that the gene that is generating the protein. But the information that we have about the annotation that is experimentally validated or that is considered as reliable is described by this blue line. So there is a big gap here, and the reason is that because providing experimental annotation with reliable support or reliable annotations requires of both experimental procedures or experimental results and manual curation of these annotations. And both of these alternatives are time consuming, and are also so expensive, even for reducing this high gap. So at this moment, we are in a key moment in which we should develop automatic prediction tools that should be able to provide us not only annotation of protein function, but also they should ensure that the reliability of those annotations is high or is comparable to those that we obtain when we assign the annotations by a means of experiment or by means of manual curation. So what are the strategies that are currently used for predicting protein functions? You can, if you explore the literature, you can find different approaches. So more or less, they can be grouped in these three different topics. So on the one hand, the most common approach is to use the amino acid sequence for using as data input for our algorithm. And the most common approach is to look for sequence similarity, or in a global way or in a local way, so just doing a blast, for instance, because it is known that proteins that are similar in sequence will have similar function or could have similar functionalities. So if you identify a protein that is really similar to your protein, you can transfer the function of the annotated protein to the unannotated protein. As I said before, this is the most common approach, but it's not the best one. Another alternative is to look for a specific feature on the sequence, for instance, for looking for specific motifs or domains that are related to a specific functionality. So if you found the specific domain or the motif in your sequence, you can assign the function that is assigned to this motif to your sequence. Similarly, you can, if you have the three-dimensional structure of your protein, you can find features or characteristics in your three-dimensional structure for assigning protein functions, or you can also use data from high-throughput experiments. One of those that is being commonly used is the protein-protein interaction network. So based on these different alternatives and approaches, a lot of different machine learning algorithms have been proposed. Some of them are based on a single algorithm, for instance, random forest, but most of the current tools are based on ensemble algorithms that they took, for instance, the prediction from a random forest and combined it with the result that they obtained using a logistic regression tool or a naive-based classification tool or a neural network, and then they combine all the predictions for obtain a final prediction of the protein functionalities. So it doesn't matter the way. The idea is that current tools are carrying out is from a protein. If we have the sequence, the structure of the information that we have, we should perform a step of feature extraction, or feature engineering, in which we define different features from the data that we have of the protein, such as, for instance, the length of the sequence, the number of or the frequency of each amino acid, or the motif, the presence of or the absence of different motifs or domains. These features are then used to fit the prediction model, and as an output, our prediction model will provide us a vector of probabilities that is telling us about the probability that each of those terms in the Gene Ontology should be annotated to the protein or not. So instead of presenting you some state-of-the-art tools, I prefer to show the results of the CAFA challenge, which is a critical assessment of different protein function algorithms, protein function prediction algorithms, sorry. So this challenge is especially designed by different groups working on the protein function prediction problem around the world. And this is aimed to provide an objective evaluation of the performance of the current tools that are available. So the idea of this challenge is, from a starting point, provide a set of proteins that do not have any annotation or any functionality assigned in UniProt at the starting point. After a period that can be, for instance, 6-7 months, the groups that are developing algorithms for predictive protein functions should provide the prediction that they obtain with their algorithms for these proteins that are annotated. And then they use annotations that were manually created by UniProt biocurators for comparing the annotations that the participants provided for the annotated protein with those that were identified during this period. And with this result, they provide a benchmark about the performance of these different methods in this set of proteins. In order to measure the performance of the algorithms, they mainly base their results using the Fmax, which is the maximum F-score that you can obtain when you average the precision and the recall of the proteins, of all the proteins, using, of course, the prediction of the model at different thresholds. So, for instance, if you have this vector of probability, in order to decide if you should assign this term to a protein or not, you should use a threshold here for converting this vector to a 0-1 vector. So they perform a whole evaluation of different thresholds from 0 to 1 with a step of 0.01, and then decide which is the max value that each method can obtain for the F-score, and this value is used for comparing the different methods. So, taking this into account, the result that they published in the last challenge that was carried out in 2017, if I don't remember well, are these. And for instance, for molecular function, the BLAST strategy that is based in sequence homology, sequence similarity, that is considered as a default or as a baseline, is achieving more or less 42%. And the best tool provided by this group is achieving more or less 62% of the Fmax among the different proteins that are provided as targets. In terms of biological processes, which as I said before, is the most complicated category because it's the biggest, sorry, is the most specific. And as you can see here, most of the tools reach at the most 40%, sorry, of the Fmax. And in several components similar to molecular function, the percent is closer to 62%. So these are the reference value that we should take into account in order to evaluate how well are the predictions that our models are able to provide us. And for this reason, I am showing you this slide. And in particular, I want to highlight, these results because I will show you some preliminary results on the biological process terms prediction. So taking into account the different alternatives that are currently available and the performance that they are achieving in the CAFA challenge, our aim or our proposal is based in two different topics. On the one hand, we want to take advantage of the current knowledge that we have of different proteins obtained from UniProt, Gene Ontology, as I said before, but also from another databases such as Reactome, KEGG, or Interpro, and also from multiomics repositories such as the Cancer Genome Atlas or the CPTAC. And in addition, in order to reduce or avoid the future engineering step on the database preparation, we propose to use a deep neural network, more specifically fully connected neural networks for modeling our problem. So more specifically, our proposal is illustrated in this figure. Here we are interested in using data from several databases, for predicting the probability of each Go term in the ontology in this way. In the first layers on the network are used for extract relevant information from each of the input that we are considering in a separate way. The next layers are aimed to combine those features that were extracted before and modeling relationships between them. And the last 4 layers are aimed to model the probability of each Go term in the ontology, being the deepest, or the last layer representing the deepest term on the ontology, or the most specific terms. So as I said before, this is a fully connected neural network with batch normalization and dropout layers. And in this case, I will present you some preliminary results from a small experiment that was performed using data from yeast. We evaluated the performance on more or less 5,800 of proteins from Uniprot KB SwissProt. In particular, those proteins have this number of annotations in these databases. These are the 3 subontologies of Gene Ontology, and these are the number of proteins that are annotated in Reactome. Reactome is a database for describing metabolic pathways in which are involved the proteins. And the experimental setup that we use for obtaining these results is described here. We use a 3-fold cross-validation scheme with 70, 10, and 20 percentages for training, validation, and testing. We use early stopping and batch size of 16, yield as activation function, a probability of dropout of 20.25, binary cross entropy as our loss function, and the Adam optimizer. And
Gabby, you have 5 minutes.
Yeah, thank you. I am finished.
Sorry.
And the result that we obtained were those. When we use only the information from protein sequence, we achieve this Fmax, more or less, is 34%. When we increase or added the information that we can obtain from the other ontologies, molecular function and cellular components, we were able to increase this Fmax to 0.46. And when we add also information from Reactome, we achieve a new improvement of this score, mainly because we were able to increase the recall of the model. So as you can see here, we are obtaining values that are closer to those reported in the CAFA paper that I showed you before. And we also identify that as more data the network is receiving, more precise and more reliable can the prediction be. So among the future works that we are planning, in one hand, we want to characterize different sets of proteins among the proteins that are available for the study. For instance, we started to look how are the proteins how are the proteins represented if we consider different sets of proteins based on if they are increasing the annotations, considering, for instance, from January to October of 2020, If they are not changing the annotations, or if those are negative, it means that the proteins do not have annotation at the beginning and at the end of the October. So we need to take into account these percentages in order to train our model in a balanced way. And we are also starting to analyze how well are each of the terms represented in the training data set. Elizabeth said something about this, because of course the terms that are more general are more represented in the data set, so their prediction is easier than those terms that are less represented on the training data set. And finally, we're also trying to evaluate or combine another deep learning architectures, for instance, recurrent neural networks or embedding autoencoders, useful for analyzing information from sequence data or even from the annotation from Gene Ontology. And finally, I want to say thank you to Diego Milone, Georgina Stegmaier from the Research Institute for Signal System and Computational Intelligence that are working with me. Thank you also to Victor Casco from the Research and Development Institute from Bioengineering and Bioinformatics, which is my co-advisor, as I said before. And thank you also to Maria from EBI-NBL, and of course to all the committee that is organizing this amazing event, and to all of you for attending my talk. So thank you.
Thank you very much, Gabi, for this lovely talk. Thank you, really. We have two questions, and there is one comment saying that how lovely your minions, and I'm sorry to be the cause of the PhD of the postdoc minion blue. No, just joking. I have three questions. Well, first one from Tayana. Great talk. How did you choose which features to choose for your data set, for your models?
Well, in our case, I do not explain a lot about our model, but our idea is to try to do not perform feature extractions. So in our case, we are only using association metrics among the different proteins. So in the model, I think I am sharing my screen. So here, the data that we are, the data that we are putting here is based on association among the proteins. We are trying to relate those proteins that are our target proteins with those that are actually annotated on the dataset. So the idea is to try to use information that we know about the proteins that are annotated to predict the functions of those proteins that are unannotated. So the only thing that we are doing is trying to associate them. For instance, PSD is protein sequence distance. So we compare the sequences of the proteins and we obtain a metric. And we use this metric as an association metrics. And these GoCC, GoMF, and Reactome, are also describing the co-occurrence of annotations among our proteins. I mean the annotated proteins and the unannotated proteins.
Okay, Gabi, thank you. The next question from Jessid. Have you checked how your method works predicting protein function in moonlighting proteins? I mean proteins that can perform more than one function.
No, at the moment we are trying to predict all the functions from all the proteins, but without looking for specific groups of proteins. But we can, I think that after finishing this first part, we will start to divide the problem, looking for different cases or different groups of proteins, as well as for different groups of terms.
Okay, thank you. Then we have another question from Eliana. Thanks for your talk. Talking about the amino acid sequence strategy, can I get more accurate from feature-based than from similarity sequence? More accurate results, I suppose.
Yeah. I get more acute from feature-based and from similarities. I think that you will have more or less the same results. We were trying and evaluating different ways for obtaining valuable features, but the differences are not significant when you consider or similarity or features. The results are more or less the same.
Okay, thank you.
It is a big challenge.
Yes. Lucia Cheme says, can you improve the predictions by using other functional data, such as that coming from post-translational modifications, binding motifs, and other short functional elements in protein sequences?
Yes, of course. I think that you can, as much data you can use, much better will be the models. But the problem is that when you are trying to predict the functionality of all the proteins, not only looking for specific proteins or in a specific organisms, you have a limited source of data. So for instance, if you are trying to predict functionalities of human proteins, maybe you have access to post-translational modification data, from transcriptomic, proteomics data, even image data. But if you are trying to analyze a non-model organism, the problem is quite different. So you can do this, but not if you are working on a tool that is trying to predict any protein from any organism. That is our case.
Yes, thank you, Gabi. The last question is, how works the Fmax? I suppose that the question goes to the difference with F1 and how do you calculate or what does it mean? The Fmax measure of the CAFA challenge.
Yes, well, it's a challenge. The Fmax is a challenge. We were discussing a lot with Georgina about it because it's like maybe it's not the best measure for this, because as all measure, it has the weakness, but it's the commonly used measure. So I think that if you are interested in develop something and to compare your results with the results that are commonly considered, you need to use it. It is helpful because it gives you an idea about how well is performing your method in comparison with the current tools. But of course, you need to complement this, analyzing at least the precision and the recall, because of course, if you can obtain a very, very nice or a very, really high Fmax, but if your precision is too bad, maybe your predictions are not so good. So of course, it will depend on what is the aim of your predicted protein functions. If you want to build a database, you need to have highly reliable prediction. Maybe if you are doing a screening study, it is okay to have a good recall and a precision not so good. So.
Okay, Gabi, thank you very much for all your answers and for your marvelous presentation.